Hello, everyone, and thank you for joining us today. I'm excited to present my work, Hilbert Curve-Based Molecular Sequence Analysis, a novel approach that bridges computer vision and bioinformatics through innovative sequence representation. This is a joint work with my colleagues from Georgia State and LUMS. Let me start with the table of content. Um, We'll begin with the motivation behind this work, then dive into the proposed methodology, followed by the experimental results, and conclude with the broader impact of our approach. The key takeaway I want you to remember is that we have developed the first universal method for converting any molecular sequence into a meaningful image representation that can then be used as an input to the deep learning model to perform classification. Um, if we talk about the challenges, so what are the problems that we are trying to solve here? In molecular sequence analysis, we face several critical challenges. Traditional vector-based embeddings, the standard approach like one-hot encoding or k-mesh-based representation they show suboptimal performance when they try to use them within the deep learning models. So why is this happening? Well, neural network, particularly CNN, struggle with the tabular data because of three main issues: feature sparsity, varying scale, and most importantly, complete lack of spatial correlations. Now, some researchers have tried image-based methods, but here's a catch. Existing approaches fail to capture proper spatial information, and current Hilbert curve methods, well, they are sequence type specific. So if you develop a method for DNA sequence, it won't work for the proteins, and vice versa. Our solution address all these limitations through what we call as universal Hilbert curve-based chaos game representation with novel alphabetic index mapping. Think of it this way. We are converting the one-dimensional sequence data into two-dimensional image that preserves spatial relationships, making them perfect for convolutional neural networks. Okay. So let me highlight our five key contributions. First and most importantly, we have developed the first universal Hilbert curve-based approach. This means that our work with DNA sequence, protein sequence, or any other molecular alphabet, no modification needed. It will work for any type of sequence. Second, we have introduced an innovative alphabetic index mapping technique that enable this universality. Third, our performance speak for itself. We achieve around 94.5% accuracy and 93.9% F1 score on lung cancer dataset, which is the state of the art as of now. Fourth, our method extend beyond molecular sequence to NLP applications, showing its broader applicability. And finally, we have conducted comprehensive evaluation across multiple datasets and deep learning architectures to ensure robustness. So let me put our work in context by discussing existing approaches. Um, on the vector-based side, we have feature engineering method like PWM2Vec, NLP-based approach like Spike2Vec or Probert, and neural network-based methods like autoencoder. The problem? These methods perform poorly with the CNN model, lose spatial information, and suffer from feature sparsity. Image-based methods like chaos game representation and FCGR perform better with the deep learning because they preserve the 2D spatial relationships and are naturally suitable for the CNN architectures. But there is a gap that we identified. Existing Hilbert curve-based method lack universality. Our approach provides a first universal solution that work across all molecular sequence types. This universality is crucial because in real-world applications, researchers often work with different type of sequences and have one unified approach is far more practical. And having unified approach is far more practical. Okay. Uh, So now, why did we choose Hilbert curve specifically? So Hilbert curve are space filling curves that map one-dimensional sequence to a two-dimensional plane while preserving spatial locality. This means that points close together in original sequence remain close together in the 2D representation. They are superior to alternatives like z-score or Peano curves because of their self-similarity and fractal properties, which maintain structure at multiple scales. For context, so z-score curves are created by interleaving the bits of the binary representation of coordinates of the multiple dimension points. For example, in a 2D space with coordinates x, y, you convert x and y to binary and then interleave their bits to form a single z order value. Similarly, if we talk about the piano curves, so they are continuous functions that map the unit interval onto the unit square or high dimensional cube, but they are not injective. This means that multiple points on the interval can map to the same point in the multidimensional space. In mathematical foundation, the mathematical foundation is elegant. For our 64 by 64 dimensional image, we use 6 iterations with 2 dimensions, given us the theta equals 2 to the power of 12, which is 4096 points, perfect for our image resolution. You can see here how the curve evolves from order 1 to order 2, maintaining its self-similarity and space-filling properties. This mathematical rigor ensures that our transformation is both theoretically sound and practically effective. Um, Let me walk you through our complete methodology. So we start with a molecular sequence. Let's say a protein sequence like ARND or a DNA sequence like ATCG. First, we apply our alphabetic index mapping. For DNA, A map to 0, T map to 1, G to 0, C to 3, and vice versa. For protein, we have 20 amino acids, so A map to 0, and so on. For each alphabet, we have like a numerical value. Next, we calculate the distance using the formula. Well, so these formulas are given in the paper, so I'll just briefly discuss here. So we have this D equal L over I over L times theta, where I is the alphabetic index, L is the sequence length, and theta is our total Hilbert curve points. Then we convert this distance to Hilbert curve coordinates using our algorithm, which involves binary representation, bit interleaving, and gray code transformation. The result, a 64 by 64 dimensional image representation that captures spatial relationships of the original sequence, ready for the CNN or any other vision model to perform classification. The key innovation here is that the universal alphabetic index mapping, it enable our method to work with any molecular sequence alphabet without modification. Okay, let me dive deeper into this crucial component. Our alphabetic index mapping algorithm is deceptively simple but powerful. For any character C in alphabet A, we return the index position of that character. Here's why this works. For DNA, our alphabet is ATGC. So adenine map to index 0, thymine to 1, guanine to 2, and cytosine to 1 to 3. For protein, we have 20 amino acids, so alanine map to 0, and vice versa. The universal property is that is what make this special. It work by any alphabet size and composition, whether you have 4 nucleotides, 20 amino acids, or maybe more than 100 smile string values. So the method adapt automatically. This flexibility is crucial for real-world applications where researchers work with diverse molecular sequences. Okay. Now let's look at how we transform indices to coordinates. So we calculate the distance d using L over I over L times theta. This normalizes the position based on the sequence length and map it to our Hilbert curve space. The coordinate transformation involves several steps. First, we convert the distance to binary representation. Then we perform bit interleaving, separating even and odd indexed bits Even bits become our raw x coordinate, odd bit become our raw y coordinates. This mathematical transformation ensures that our sequence elements are distributed across the 2D space in a meaningful way that preserve the original sequence structure. The crucial step in our process is the gray code transformation. Why do we need this? So gray codes minimize bit changes between successive values, which is essential for preserving spatial locality in our Hilbert curve representation. We apply XOR operation and right shift to convert our raw coordinates to gray code, then apply inverse gray code to get our final coordinates. This ensures that um, this ensures that sequence elements that are closer together in the original sequence remain close together in our 2D image representation, a property that's crucial for CNN performance. So here is our core algorithm that ties everything together. So we take the distance, convert into binary, separate even and odd bits, apply gray code transformation, and refine the coordinates to get our final XY position on the Hilbert curve. This algorithmic approach ensures consistency and reproducibility across different sequence types. So let me describe our experimental setup now. We test our method on two anti-cancer peptide datasets, breast cancer with 949 sequences and lung cancer with 901 sequences. So these are four classification problem ranging from very active to virtually inactive peptides. The sequences are relatively shorter, 5 to 38 amino acids with average length around 14 to 20 amino acids, which make it a challenging classification task because there is very less margin to capture the patterns in the sequences. So we compare against comprehensive baseline, including traditional vector-based method like one-hot encoding to modern approach like sequec, which is a LLM-based approach for the sequences, as well as we also compare it with the image-based model like Spike to CGR and FCGR. For deep learning architecture, we tested everything from a simple one-layer CNN to sophisticated model like ResNet 50 and EfficientNet. So our training setup used standard practice like 80% for training, 20% for testing. We use the ADAM optimizer and did the hyperparameter tuning using the validation set, which is basically the 10% data that we get from the training set. We evaluated performance using the standard classification metrics like accuracy, precision, recall, F1 score, and ROC. We use both weighted and macro F1 score to account for the class imbalance and Meyer training runtime for computational efficiency and assessment. All experiments were conducted on standard hardware with proper cross-validation for hyperparameter tuning. So let's look at our results starting from the breast cancer data. Our results, our method achieved 89.5% accuracy, as you can see in this bold blue, compared to the best baseline, which is 86.3 for the FCGR with one layer CNN. So what's particularly interesting is that our simple one layer CNN performed best. This supports the Occam's razor principle that simple solutions are often better. We see significant improvement over F1 score. If we talk about the number, so it's 0.52, like the 52% versus the 49% in the for the baseline, which indicate better handling of the class imbalance. Now, how notice that how traditional vector-based method like autoencoder perform reasonably well, but our image-based approach consistently outperform them. So the lung cancer data results are even more impressive. 94.5% accuracy, the highest among all methods tested, and 93.9% weighted F1 score, which represent exceptional classification performance. We outperform sophisticated baseline like FCGR with three-layer CNN that achieve 93% accuracy. So that's a 1.5% improvement, which is substantial in this domain. Again, our simple one-layer CNN architecture performed the best, reinforce that our image representation is so effective that complex architecture are necessary. This consistent performance across both datasets demonstrate the robustness and reliability of our approach. These results suggest that our method could have significant practical impact in drug discovery and cancer research. So let me share some key insights from our analysis. First, our method consistently outperform all baselines on both datasets. This isn't a fluke, it's a systematic superiority. Second, this simpler is indeed better, or one layer CNN achieve the best results, which suggests or image-based representation capture the essential information so effectively that complex architecture actually hurt performance. Interestingly, very deep network like efficient net show poor performance, only 6 to 8% accuracy, indicating they are overcomplicating the problem. So why does our method work so well? So Hilbert curve preserves spatial locality or universal mapping work with any alphabet, and we maintain information preservation through bijective relationships. The mathematical guarantees of bijective mapping ensures each character map to a unique Hilbert curve point, preventing information loss. So beyond accuracy, computational efficiency is crucial for practical applications. Our method is 2 to 3 times faster than competing image-based methods. For the breast cancer data, we complete training in around 2000 seconds compared to over 5000 seconds for the FCGR. So this efficiency advantage make our method suitable for real-world deployment where computational resources and time constraint matters. The scalability is also excellent. Training time scale reasonably with model complexity. So let me summarize our key algorithmic innovation. First, universal alphabetic index mapping that works with any molecular alphabet. Second, or distance calculation that normalize position and map to the Hilbert curve space. Third, gray code transformation that preserves spatial locality. These features combine to provide bijective mapping with no information loss, spatial coherence where nearby elements remain close, and fractal properties that maintain structure at multiple scales. The fixed 64 by 64 resolution provides standardization while maintaining sufficient detail for classification. So here is our performance summary compared to the state-of-the-art method. We achieve an average improvement of 2.6% over the best existing method with consistent performance across both datasets. Our competitive advantage include universal applicability, computational efficiency, and strong theoretical foundation with mathematical guarantees. This represents a significant advantage in the field of molecular sequence analysis. The implication of this work extend far beyond our specific experiments in bioinformatics. This could revolutionize drug discovery by helping identify therapeutic peptides more accurately. For disease diagnosis, it enables better biomarker classification. In genomics, it provides a new way to analyze DNA and RNA sequences. But the impact goes beyond biology or method can be applied to natural language processing, time series, or any sequential data with spatial patterns. We have created a methodological innovation that bridges computer vision and bioinformatics with a universal framework applicable to diverse molecular alphabets. Future direction include integrating integration with transformer architecture with multimodal learning approach, which could further enhance the performance. Okay. Uh, so let me just conclude our key achievements. So as we talk about the superiority in terms of the predictive performance, we develop a universal method applicable to any molecular sequence with better computational complexity. The scientific impact is significant. We are bridging computational vision and bioinformatics through novel sequence representation with strong theoretical foundation, practical significance for drug discovery and disease detection. We have created an extensible framework that open up numerous future research directions. With that, so I'll stop here. Thank you for your attention, and I'll be happy to take any questions. You can email me if you have any question. Unfortunately, I couldn't be there in person to present my work, but I would be happy to answer any question over the email. Thank you.